All right, guys. So we're now going to be covering topics point 4 to point 7. And this is pretty much, again, the course at a glance. So for this presentation, we're going to hit pretty much all the topics that we have left. So carbohydrates, lipids, nucleic acid, and proteins. All right, so this is our topic, biological macromolecules. And again, we're going to be covering the four major macromolecules. We're starting off with carbohydrates. Let's start off with our carbohydrates. Carbohydrates include sugars, like simple sugars that are sweet to taste, and also more complex carbohydrates are polymers of sugars, which are like, for example, starch, and they're starchy and a little bit more stainy when you actually taste those. Um, the structure of carbohydrates. Here's an example of the structure of carbohydrates. If you look at the structure, you should be able to identify these two functional groups, which are a key part of the structure of carbohydrates. So the two functional groups that are involved in carbohydrates, and please note this, we have the carbonyl group and the hydroxyl group. I would advise that you pause the video right now and try to identify these two functional groups on this structure right here. All right, so the carbonyl group is the carbon double bonded to oxygen, and the hydroxyl group is OH groups. So wherever there's an OH group, those are hydroxyl groups. Carbohydrates is made up of carbons, hydrogen, and oxygen only. There's no other element that is involved. So this is our CHO, and you guys should recall this from pre-AP bio. There are different subcategories or classes of carbohydrates. We have monosaccharides, which are simple sugars, usually monomers. Um, these molecules are usually formulated in a way that is made up of this specific ratio. And by ratio, what I mean is that within the monomer, within these monosaccharides, there is one carbon to every two hydrogens and one oxygen to every two hydrogen. So the ratio is one carbon to two hydrogens to one oxygen. If we increase the number of hydrogens to, for example, if we increase the number of hydrogens to 4, then that the ratio is also going to increase. So that's going to be 2 carbon to every 4 hydrogen and 2 oxygen to every 4 hydrogen, if that makes sense. So that is the ratio with monosaccharides. The most common monosaccharide is glucose. So glucose is a monosaccharide. It's a simple sugar. And glucose is used. So again, remember, we're also trying to determine function of these macromolecules. What is glucose used for? Glucose is used for fuel. So glucose is the primary molecule that is used in cellular respiration. So when during metabolic processes in which energy is needed, glucose is that molecule that is primarily used. Can also serve as building blocks for amino acids or as monomers for disaccharides. And disaccharides is another category of, it's rarely ever mentioned, but we also do have disaccharides. So we have monosaccharides, meaning this molecule or this compound is made up of one sugar monomer. And then we also have disaccharides, in which the compound is made up of two sugar monomers. And then we have polysaccharides, and polysaccharides is when we get to more complex carbohydrates, so like our starch, and those are polysaccharides, in which it is made up of many monomers of carbohydrates. And then pretty much this is just to reinforce what I just said. So disaccharides, two monosaccharides joined together, they're covalently bonded. And in order for us to get two monosaccharides to join together, what type of reaction would cause those two monomers to covalently bond together to form a disaccharide. So think about it, and that's from our previous lecture. So the answer that should have come to you was you have to dehydrate. So water has to be removed in order to get these two to bond. Whenever we have two monosaccharides coming together, two simple sugars, it's usually a glucose and a fructose, and it usually forms what is called sucrose. And sucrose is a disaccharide, which means it's made up of two sugars. So glucose, fructose, and plants can transfer this from the roots to the leaves, and it can be broken down to pretty much be used in cellular respiration. Polysaccharides, I also mentioned what polysaccharides are. Polysaccharides are many monomers or many sugars come together, and those sugars are all going to be covalently bonded together using dehydration and synthesis. Polysaccharides, the function of polysaccharides is for storage in plants. So polysaccharides are mostly used for storage. So starch is stored in plants as more complex carbohydrates, and when the plants are ready to actually use the use the monomers for fuel for cellular respiration, it breaks down the starch. So this allows for plants to store excess glucose by pretty much causing the glucose molecules or monomers to form these complex polysaccharides. Animals cannot store glucose, so we're incapable of storing glucose. But whenever we have excess glucose, we convert our glucose to glycogen, and that is how we store. So glucose should never be stored in our blood because that makes us have high glucose levels in our blood, which is not good for our physiology. So therefore, we convert our glucose to glycogen. And this is stored in the liver, and glycogen can be converted to glucose when our body needs that glucose. Here's something that's also very important with carbohydrates. Carbohydrates form structural compounds such as cellulose. So cellulose in plants. So we've heard of, I'm pretty sure you guys have heard of cellulose cell wall. So plant cell wall is very tough, very rigid structure, and that is because it is made up of this cellulose cell wall. So those are some of the functions of carbohydrates. Oh, and also chitin. So this word is actually pronounced chitin and not chitin. So it's pronounced chitin, and chitin is pretty much what forms the exoskeleton in arthropods. So in insects, that tough outer layer is made up of chitin, and chitin is a type of polysaccharide. So these are structural carbohydrates or structural polysaccharides. All right, here's a practice question for polysaccharides, sorry, for carbohydrates in general. So it says here you are given an unknown monosaccharide to identify in the lab. The only clue you are given is that it has 4 carbons. You, being an excellent AP Bio student, figure out the formula and name it immediately. What is the unknown monosaccharide? So you have figured out what the formula is. If it has 4 carbons, and we know that carbohydrates are made up of only carbon, hydrogen, and oxygen. It has 4 carbons. So remember the ratio. If it has 4 carbons, it is always going to be 1 carbon to every 2 hydrogen and 1 oxygen to every 2 hydrogen and 1 carbon. So knowing that ratio, you should have come up with this formula. And then you can go ahead and look up what type of molecule that is. You're not expected to know this carbohydrate is, or to be able to name the carbohydrate, but I just added that to the question. So this is pretty much based on the ratio. So it's 1 to 2 to 1. So the same amount of carbon should be the same amount of oxygen, and then hydrogen should always be times 2 whatever the carbon or the oxygen is. All right, that was carbohydrates, and now we're into proteins, guys. Proteins are my favorite macromolecules. I think proteins are just super interesting. All right, so formations of proteins. Proteins form from amino acids um, covalently bonding together to form peptides, and then peptides are pretty much even more amino acids bonding together to form very, very long chains, and we call those polypeptides. So whenever you hear the word peptides, I want you to always think of proteins. And then what are the monomers of protein? The monomers of proteins are amino acids. So proteins are molecules containing polypeptides, which makes them polymers of amino acids. So amino acids are the simplest or the building blocks of proteins. Proteins are folded in 3D shapes, so three-dimensional shapes, and the shape of the protein determines its function. Now, before we go there, let's talk about what protein is actually made up of. So protein is comprised of CHON, so the CHON. So protein is made up of carbon, hydrogen, oxygen, nitrogen, and some proteins, depending on their functional group, is also made up of sulfur. So there are some types of amino acid that is actually made up of sulfur. Um, I am going to go ahead and I'm going to put a box around this bullet point right here because this is so crucial to our understanding of proteins, right? Not a perfect box, but a box nonetheless. So protein are folded into 3D shapes, and these are intentional folding. There's specific foldings for all these polypeptides. The shape of the protein, based on how it is folded, is what determines how the protein works. So shape determines function. Shape determines function. I want you guys to always remember that when we talk about proteins. All right, so proteins are made up of amino acids. Here is the chemical structure of an amino acid. There are 22 different amino acids in living things. There are 20 functional amino acids. So amino acids that actually do something. They actually code or they can be used to synthesize specific polypeptides. So molecules that have molecules that have amino group and a carboxyl group are amino acids. So remember what these functional groups are. So amino group is the group that has one nitrogen and two hydrogens. And I want you guys to recall that from our slide where we discussed different functional groups. And then carboxyl group, remember carboxyl group is a combination of carbonyl and hydroxyl, in which we're going to have a carbonyl group covalently bonded to a hydroxyl group. And that's what gives us our carboxyl. So amino acid is pretty much a short carbon backbone. So here we have our hydrocarbon, which is our carbon and hydrogen. And that group has two functional groups. So we're going to have our amino group and our carboxyl group. And here's a very, very, very important group, guys. These are called or R groups. And our R groups are variable chains. And we put R here to pretty much symbolize that for different types of amino acids, the reason why we have 20 different functional amino acids because there are 20 different R groups that could possibly be added to amino acids. And each R group gives protein a unique function. And each R group also determines the shape of the protein. So that 3D folding of the protein. So how does the polypeptide actually fold is determined by what are the R groups that are made up, what are the R groups that actually comprise this protein. And here's an example. So examples of different proteins. So if you look at the structure of these two proteins, you're going to find that everything is the same. So here we have, what group is this? This is our amino group. This is our carboxyl group. And then here we have our carbon backbone. And then there is another element that is added, or sorry, another compound that is added, which is unique. And then if we go over here, again, we can identify amino group. And this is our carboxyl group. And this is our carbon chain, or this is our carbon backbone. And then here, as opposed to having this this methyl group, what do we have? We have a hydrogen. Now, these two amino acids, so this is a monomer and this is a monomer. However, because the functional groups, or the R groups, are different for these amino acids, it means that these amino acids now have different functions, or they're different types of amino acids. Still amino acids, but they're different types of amino acids. And based on this, this will determine how this amino acid functions with other, or how it interacts with other amino acids. So these are the R groups, or the R chains. So unique side chains are R chains. And based on these chains, whenever we talk about glycine or alanine, these are different amino acids. And what makes them different? The side chain. Everything else is the same. Amino group, carboxyl group, the carbon backbone, everything is the same. The only thing that's different with amino acids are these R chains, and that's what gives them their unique properties. And that's when scientists start naming them as different amino. All right, a little bit more about amino acids and their side chains. So each amino acid, abbreviated AA, has unique side chains, and side chains are the same as the R groups. So what it does, it pretty much gives these amino acids like unique properties. So physical and chemical properties of these amino acids are now unique depending on what side chains they have. And what are some of these unique physical and chemical properties? Here they are. So based on the side chains that the amino acid has, it could cause the amino acids to behave nonpolar, in which it is hydrophobic, water-faring, or it could cause it to be polar, in which it is water-loving, it's hydrophilic, and it interacts with water. Or the amino acid could behave like an ion, so it's charged, whether positively or negatively, and this also is hydrophilic behavior. So side chains interact, and side chains will interact with other side chains. And that is what determines how the proteins fold. So that 3D folding of the protein is based on what side chains are in that long chain of amino acids or polypeptides. And based on how it is folded, which is a shape, is what determines how the protein actually functions. All right, for this slide, we're gonna get into how are these polypeptides formed. So in order for monomers to become polymers, remember the type of reaction that will occur is always going to be dehydration synthesis or dehydration reaction. So water has to be removed in order for monomers to covalently bond with other monomers to form these polymers. And protein is no exception. So amino acids will bond with other amino acids as a result of dehydration reaction or dehydration synthesis. Now, for proteins, it occurs in a specific way. So I want you guys to try to recall the structure of an amino acid. What are the groups of the amino acid? So remember, amino acid is made up of the carboxyl group and the amino group, and then we have of course the carbon hydrogen backbone. So that hydrocarbon backbone. So we're going to focus on the two functional groups that are in all amino acids. In order for dehydration synthesis to occur, in which we're building long chains of polypeptides, the carboxyl group of one of the amino acid must be positioned right next to an amino group of the other amino acid. So And in doing so, here's what will happen. So here we have alanine and glycine. I'm going to put a circle, or I'm going to attempt to put a circle around this group right here. So this is a group, a functional group from one of the amino acids, and then here we have a functional group from the other amino acid. And I want you to tell me what these two functional groups are. So which is which? So remember when we have one nitrogen and two hydrogens, that forms the amine group. And then whenever we have to carbon double bonded to an oxygen, and we have an hydroxyl group, that is our carboxyl group. So let's go back to what it says here. In order for us to have the dehydration synthesis in which we're forming long chains of amino acids, which are called polypeptides, we have to have the carboxyl group of one amino acid next to the amino group of the other amino acids. So here we have them side by side. And remember, dehydration synthesis is the same for every single molecule, for every single compound that is going to be covalently bonding. We have to remove an entire hydroxyl group from one to give us one hydrogen and one oxygen, and then we need one hydrogen from the other group. So we're going to take the entire OH, that is the hydroxyl group, from the carboxyl group, functional group, and then we're going to be taking a hydrogen from the amino group, and that's what gives us our H2O. Once we have removed these two from each group, now this amino acid is unstable, this amino acid is unstable, and therefore, in order to gain stability, the carbon from here and the nitrogen from here, or the other hydrogen from here, needs to covalently bond with that carbon. So that is pretty much what's going to happen. And that is what we have. So here we have how they bond. And this is as a result of dehydration synthesis. All right, so polypeptides are pretty much long chains of amino acids. So the sequencing of amino acids is what determines that 3D shape. So remember, there are 20 different functional amino acids. So how are they sequenced? So what amino acid is first, and then what amino acid is next, and then what amino acid is next. That's what we mean by the sequencing of the amino acid. And based on how the amino acid is sequenced, it is what will determine how the protein is folded. And pretty much what that is saying is that if you have an assembly line of, like, there are 20 different options for amino acids, and we're forming a long chain of polypeptide, which is going to be the first amino acid, and then what type of amino acid is going to be next, and then what type of amino acid is going to be next, and then what type is going to come next. And as you're assembling amino acids, you also have to bear in mind that, hey, these amino acids have unique properties based on the different R groups or R chains that these amino acids have. So while they all have the amino group and the carboxyl group in common, remember that there are different R chains. So as you're sequencing them, the R chains are going to be interacting with each other. And based on how the R chains interact with each other, that is what determines how the protein actually folds. So remember, there are some parts of the proteins that are going to be hydrophobic, in which they're water faring, so they're going to want to fold into the inner core of the protein, in which they can hide themselves from water. There are some parts that are going to be a lot more hydrophilic, so they're okay with being exposed on the outside. So that is what determines how the protein actually folds and forms that 3D structure. So it says here, when a polypeptide twists and folds because of the R chain interaction, like I just explained, it forms what is called a protein. And here's a we probably should have done this in class, but it's like a think, pair, share. So how is the unique sequence of the amino acid determined by the polypeptide? So what determines the sequence of amino acids? So what determines the order in which these amino acids are assembled is genes, and we did that in six in unit 6. Of proteins. So what are some functions of proteins? Proteins, they form antibodies. Antibodies are protein structures. They're chemical compounds that pretty much play a huge role in immunity and defense. So how can we fight against pathogens and invading organisms? We produce antibodies, and antibodies are made up of proteins. Also enzymes. So proteins form enzymes. Enzymes are proteins. And proteins are and enzymes are responsible for carrying out metabolic reactions. Specifically, they're biological catalysts, so they speed up metabolic reactions. Proteins also functions as messengers, so they can transmit signals across long distances, for example, in the form of hormones. Proteins also provide structural support. So like our muscles are mostly protein, and there are other structures within our body, especially in our skin, or that are more structural structures, and therefore they are more physical structures. And then proteins can also be used in transport and storage. So proteins can bind to carry small atoms through the body. So they're responsible for pretty much carrying things from one place to the next. All right, so here's something very important that we need to note. Levels of protein structures, guys. So this has to do with the actual physical structure of the protein. The first level is called the primary level. And this is a linear chain of amino acids. So this is pretty much the sequencing of the amino acids based on how the genes were transcribed and translated. And during the translation process, these are the proteins that the tRNA brought in this specific order, right? So this is our first protein, followed by this one, followed by this one, and they're all covalently bonded together. So this is our sequence of amino acids, right? Now, this sequence, the very simple basic structure of protein, is just a linear structure. So when we talk about the primary folding or the primary structure of protein, it's simply just like in what order are these amino acids assembled. They're determined by genes. Um, the primary structure is what determines the next level, which is the secondary structure. So I want you guys to always think, remember, proteins, each amino acid, each of the 20 amino acids, they're unique because of the R chains. So for example, glycine has an entirely different R chain than serine has. And therefore, glycine has different chemical properties than serine does. Maybe glycine is hydrophobic, and serine, on the other hand, is more hydrophilic. Or maybe serine has a is positively charged, and glycine is negatively charged. So that will determine how they fold. And also, what if they have the same charge? If glycine and serine are both positively charged, they're going to repel each other. So they're not going to be folding on each other. They're going to be folded in a way that they're pretty much avoiding each other. So the position of the amino acids is pretty much what determines how the protein folds. Now, the secondary level, which is determined by the primary level, is pretty much when proteins are folded, or they're beginning to fold in a way that they form coils due to hydrogen bonding between the peptide backbones, right? So, and when we talk about the peptide backbones, we're talking about the parts of the protein that is not the R groups. So, based on hydrogen bonding between those peptide backbones, they can form what are called beta pleated sheets. in which we have hydrogen bonds between polypeptide chains lying side by side, or we could have what is called the alpha helix, in which we have hydrogen bonding between every fourth amino acid. So beta pleated sheets and alpha pleated sheet, they are similar in the sense that this is a folded pattern based off hydrogen bonds between the peptide backbones. Beta pleated sheets are formed if the hydrogen bonds are formed between the polypeptide chains that are lying right next to each other, side by side. That forms beta pleated sheets. We get alpha helix if hydrogen bonds occur between every fourth amino acid. So that is the difference between the two. So where are these hydrogen bonds forming is what determines if they're beta pleated sheets or if they are alpha helix. So we have our primary level, we have our secondary level, and now we have our tertiary level. So we're building up to the tertiary level of proteins. Now the tertiary level is when protein starts to become 3D, three-dimensional. Now this is when our interaction between our R chains come into play. So tertiary, our 3D folding, is due to interactions between those side chains or these R chains. And remember, there are different ways in which R chains can interact based on what these R chains actually are. So these are reinforced by hydrophobic interaction and disulfur bridges of these R chains. The covalent bonds form between sulfur atoms of two cysteine monomers. So if we have disulfur bridges, it means that there are two cysteine monomers that will have to be interacting in a way that they're forming these bonds, and it's sulfur from one cysteine with the sulfur from another cysteine forming what are called disulfur bonds. And then also we have quaternary, we have the quaternary folding, and quaternary folding is as a result of two polypeptide pretty much folding onto each other. And for this diagram, I use two different colors. So this, for example, this is a long chain of polypeptide, or a long chain of amino acid that forms a polypeptide. And then the darker blue is another long chain of polypeptides. So these are two separate polypeptides now coming together to fold on each other. And that is our quaternary folding. And again, the way a protein is folded, so the shape of the protein is what determines its function, sorry. So shape determines all right, guys, so here's a question. I want you guys to read the question for yourselves, pause, and try to determine what the correct answer is for this question. And then you can unpause the video and we're going to go over the question. If you think you already got this, you can just move on. All right, so this practice question is about cystic fibrosis, which is an actual real genetic mutation that resulted from the deletion of 3 nucleotides. So the deletion of 3 nucleotide bases, and you should know what those are. The cystic fibrosis transconductance regulator gene is now affected. So this gene codes for transmembrane proteins that regulates ion channels in epithelial tissue. So if 3 nucleotides are deleted, then it means that this will result in a frame shift mutation in which the way the nucleotides are going to be read will be affected. And also remember, 3 nucleotides code for 1 amino acid. So it's also going to be coding for different amino acids. So whenever transcription occurs, those 3 are deleted. So therefore, the wrong transcript will be produced. And also, if the wrong transcript is produced, then it means that the when translation occurs inside the ribosomes, the amino acids are not going to be assembled in the correct order in which they should be assembled. So, what structure of protein is going to be affected? And you should think primary structure. So the primary structure of protein is going to be affected, in which, remember, the primary structure of protein is pretty much the sequence of the amino acid. So what order are these amino acids being sequenced in? The order will determine how the amino acids fold, because remember, these side chains will be interacting with each other to determine the 3D structure of protein. So if the sequence of the amino acid is affected, it affects how the protein folds. So it affects the 3D shape or the structure of the protein. And if it affects the shape of the protein, it means that it also affects the function. So remember, the shape of the protein is what determines its function. So protein did not fold correctly, and therefore protein does not work correctly. And simple things, well, are not simple, but small, minute errors such as this could lead to life-threatening diseases such as cystic fibrosis. All right, now we're going to discuss nucleic acids. So nucleic acids are polymers made up of nucleotide monomers. So nucleic acid is composed of several nucleotides forming these structures, and nucleotides are bonded together to form longer chains of nucleic acid. This is something we discussed in topic 6.1. So again, the function of nucleic acids in general is to transmit, and to express hereditary information. So when we talk about nucleic acids, we're talking about genetic information. So this is our DNA or RNA. There are two forms of nucleic acid, as you should already know. We have deoxyribonucleic acid, which is our DNA, and we have ribonucleic acid, which is our RNA. Components of nucleic acid. So nucleic acid is made up of nucleotides. So nucleotides are the monomers. Several nucleotides form polynucleotides, and many polynucleotides will form what we call nucleic acid. So monomers, and then we have pretty much the polymers, and then those polymers will lead to what we call nucleic. So nucleotides, again, these are the monomers, and as you guys should recall from our previous topic unit 6, sorry, 6.1, nucleotides are made up of three parts. We have the nitrogenous base. We have a 5-carbon sugar, or called a pentose sugar, and we know that the 5-carbon sugar specifically is ribose. And then we have a phosphate group, and now we know a little bit more about phosphate groups. Phosphate groups is one of the functional groups, sorry, and that is a phosphorus bonded to a few oxygens. So these are the parts of our nucleic acid. And here's something we should also know. In a polynucleotide, each of the monomer, which is a nucleotide, has only one phosphate group. So nitrogenous bases. So the nitrogenous base, there are 5 different types of nitrogenous bases. So we have our cytosine, thymine, uracil, adenine, guanine. Uracil is found only in RNA, so ribonucleic acid, and thymine is found only in DNA, D-ribonucleic acid, and this is prior knowledge. We're going to be grouping these nitrogenous bases into 2 groups, and you should also know the 2 groups in which these nitrogenous bases are categorized. So there are 2 types of nitrogenous bases. They are the pyrimidines and the purines. The pyrimidines are the smaller ones that are made up of only 1 ring, and the purines are the larger nitrogenous bases, which are made up of 2 rings. So from this diagram here, showing the chemical structure, how the molecule is configured, the bigger structures are our purines, which have 1, 2 rings, and our pyrimidines have 1 ring. And here they are. And again, this is prior knowledge. So you should know at this point what groups each or which group each of the nitrogenous base belongs to. A nice way to remember, and I mentioned that in the Unit 6 lecture that we did, for the smaller bases, sorry, for the smaller nitrogenous bases, there are three of them. One, two, three. And you can use the acronym CUT, which is C for cytosine, U for uracil, and T for thymine. So it's as if these smaller ones, they're smaller, they have their bases were cut, or the number of bases is cut by one. It's silly, but that's just a nice memory gem for you guys to remember which ones are the single ones. All right, so now we're going to talk about the 5-carbon sugar, or the pentose sugar. So we should know that the sugar is called ribose, and the sugar is slightly different in DNA than it is in RNA. The sugar in DNA is called deoxyribose, and the sugar in RNA is just called ribose. Now, the reason why in DNA it is called deoxyribose is because in our RNA, so let's look at our diagrams here. So this is DNA, and if you look at the structure, if you were to compare these two, you're going to find that they're pretty much identical except for this hydroxyl group. So this hydroxyl group, this OH group, is missing the oxygen. So it has been deoxygenated, while in our this ribose sugar, which is found in RNA, has two hydroxyl groups. So in DNA, there is one hydroxyl group, and in RNA, there are two hydroxyl groups. And where that second hydroxyl group would be in DNA is not a hydroxyl group because the oxygen has been removed. So that's why we say it is deoxy, D meaning oxygen removed. So let's talk about the third part of our nucleotide, which is a phosphate group. And let's look at the chemical configuration of the phosphate group. So the phosphate group is right here. It is made up of a phosphorus bonded to 4 oxygens. And one of the oxygens, the phosphorus, shares a double bond with that oxygen. So this is our phosphate group. And we know that phosphate groups are functional groups. So here we have the phosphate, and the phosphate group is always going to be added to the 5' end, or the 5th carbon in the sugar. So here we have our sugar. This is our ribose sugar. And if we were to count carbons, we have 1, 2, 3, 4, 5. So wherever there is the lines are intersecting, that is a carbon, and that's just how it is represented in this in these diagrams. So carbon 1, 2, 3, 4, 5. This 5th carbon is where the phosphate group is added. And whenever we're talking about the orientation of DNA or RNA, we say the 5' end or the 3' end. The 5' end is this region where the phosphate is, and we can always determine where this is, because if it is a region that is sticking up or if it's a region that is facing down, then we get an idea as to how the nucleotides are oriented in the nucleic acid. All right, so let's talk about the formation of polynucleotides. So polynucleotides are formed as a result of several nucleotides coming together. And what I have circled here is one nucleotide, and we're going to try to identify the parts of the nucleotides. So this is our phosphate group. This is our sugar, and this would be our nitrogenous base right here. So with these 3 parts, this makes up one nucleotide. And as you can see, this nucleotide is covalently bonded to another nucleotide, and then that nucleotide is covalently bonded to another nucleotide. So within this structure right here that we're looking, we can see that there are 1, 2, 3 nucleotides. And we can also see that the first and the second one, the nitrogenous bases, because they only have one ring, that has to be our pyrimidines. And then the third one has 2 rings, so that has to be our purines. I hope you guys are making notes of that and you are able to explain those things. All right, so let's talk about polynucleotides. So polynucleotides are formed as a result of the phosphate group. So here we have our phosphate group. Our phosphate group is connected to the sugar. So it's always going to be a connection between phosphate groups and the sugar group, phosphate group and the sugar group. And it's going to form that chain. So here we have our phosphate group attached to the sugar. And for this region right here, we have the phosphate group attached to the sugar. So that is how the polynucleotides are formed. It's always going to be phosphate covalently bonding with the sugar group. And then nitrogenous bases are pretty much unique. So it could be any of the base, cytosine, thymine, uracil, if it's RNA. So whatever bases, and the sequence of the bases pretty much determines the amino acid sequencing. So what amino acids will be assembled once that codon is formed. And it also gives a sense of directionality. So where is the 5' end, where is the 3' end, and so on. So again, so here's the key takeaway from this slide. Um, in order to form polynucleotides, there has to be covalent bonds between each nucleotide. So, and the nucleotides are covalent bonded to nucleotides are formed in which phosphate groups are covalently bonded to the sugar groups. Now, in order to form a double-stranded structure, the bases, the nitrogenous bases, will form intermolecular bonds between other nitrogenous bases, and those form as a result of hydrogen bonds. And we're going to get into that part of it. All right, so we're going to talk about DNA as an example of forming that double helical structure. So DNA is double-stranded. RNA is not double-stranded, but DNA is. So whenever the polynucleotide structure is formed, and we know how polynucleotide structures are formed from our previous slide, now we're going to talk about DNA specifically, in which two polynucleotide strands come together. Now, when 2 polynucleotide strands are coming together, it forms a double helix, and each strand has to be anti-parallel to the other strand. How are these 2 strands held together? They're held together by hydrogen bonds. So hydrogen bonds will occur between the nitrogenous bases. And depending on the bases, it depends on what bases are being joined together, and we should know the base pair rule. It could be 2 hydrogen bonds, or it could be 3 hydrogen bonds, depending on what the base is. So for example, cytosine always pairs with guanine. Between cytosine and guanine, they are held together by stronger intermolecular forces, because there are 3 hydrogen bonds between these 2 bases specifically. And if you look at my example right here, 1, 2, 3 hydrogen bonds will hold these 2 bases together in order to form that double strand. However, for thymine and adenine, it's 2 hydrogen bonds that hold those together. It is easier, not easy, but relatively easier to separate thymine from adenine than it is to separate guanine from cytosine, because there are more hydrogen bonds between guanine and cytosine. So that's just a fun fact. Now for RNA, RNA forms polynucleotide structures. However, the polynucleotide structures are not joined to another polynucleotide structure by hydrogen bonds, because RNA is single-stranded. So RNA can take various shapes. The RNA can coil on itself in various ways. But however, it's not going to be it's not going to be joining with another strand to form that. All right, so here are 2 quick concept check questions, super easy questions. I'm not going to go over the question in the interest of time, because I want to go to lipids. So here are the questions. Pause the video and try to answer these questions. I trust that you guys can do this, because we covered this sufficiently in 6.1. All right, so these are the questions. Try to answer these questions. If not, talk to me during class if you're having any difficulties. But we're going to move on to our next macromolecule, our very last one, which is lipids. Almost done. So here we are. All right, so as discussed earlier, lipids do not include polymers. They're very small, so they're generally very small, especially in comparison to the other three macromolecules that we discussed. And oftentimes they're not even considered to be macromolecules because of their size. Some very important things to note about lipids are non-polar, and we should know what polarity means, and we should know what polar molecules are and what non-polar molecules are. So lipids are non-polar, which makes lipids pretty interesting in terms of their chemical behavior characteristics. So if they're non-polar, they're not going to be interacting with polar molecules. And as a matter of fact, it's going to form like a repulsion with polar molecules. So lipids, the molecules have no charge. And there are three types, or there are three categories of lipids. So we have fats, we have phospholipids, and we have steroids. So these are the three types of lipids, and we're going to be discussing each type. All right, so let's start off with fats as our first type of lipids. So I'm going to tell you a few things about fats, a few things that are very, very important. So if you're taking notes, I would advise that you do ensure that you capture some of this information. So fats are composed of two things. So there are two chemical regions that are very important in fats. That's the glycerol end and then the fatty acid end. The glycerol end, and there's just one glycerol to every three fatty acids. So the glycerol is classified as an alcohol because it is made up of the hydroxyl groups. And hydroxyl, we know, is a functional group. And if you didn't know this, the hydroxyl groups will give any molecule alcohol properties. Fatty acids are long carbon chains, so it's long chains of carbon and hydrogen. So they're like hydrocarbon chains. Um, in which we have a carboxyl group at the very end. And remember what a carboxyl group is. Each glycerol is attached to three fatty acids, and those attachments are where they attach, or the bond between the fatty acids and the glycerol is called ester linkage or ester bonds. And these, when we talk about ester bonds, it is bonds. These are bonds between hydroxyl groups and carboxyl groups. So those are ester bonds. All right. So fats. Fats can be classified or placed into two groups. We have saturated fats, or what is called saturated fatty acid, and what is called unsaturated fatty acids. And here's the difference between the two, and this is very important. For saturated fats, it means that there is so many hydrogen in the hydrocarbon structure that there is no need for carbon to be double bonded with carbon, because there are enough hydrogens to satisfy carbon's stability. So there is enough hydrogen to actually share electrons with carbon, so carbon does not need to double bond to itself. So think of the word saturated as there are many or plenty or enough hydrogens. So no carbon double bonded to carbon. The opposite of this is unsaturated fats, in which there is not enough hydrogen, and therefore carbon will share electrons with other carbons in order to satisfy its stabilities, in order to become stable. So there's usually one or more double bonds of carbon with carbon in unsaturated fats. All right, now let's talk about phospholipids. And this is the one that you guys should be familiar with, because you've been learning about phospholipids whenever we talk about the structure of cell membrane. So the cell membrane is made up of phospholipids. So this is the structure that we're talking about. So phospholipid is made up of a phosphate group. So whenever you think of the phospho part of it, we're talking about a phosphate group. And we know that a phosphate group is pretty much phosphate bonded to four oxygens, in which they form phosphorus will form a double bond with one of the oxygen. So for phospholipids, Phospholipid is made up of two fatty acids attached to a glycerol and a phosphate. So there are three regions. So here we have the phosphate, here we have the glycerol, and we have two fatty acids instead of one fatty acid. One of the fatty acids tail will be saturated. So remember what it means to be a saturated fat. It means that there is enough hydrogen in which carbon does not need to double bond with carbon. The other tail is going to be unsaturated, in which carbon is double bonded to itself at least once in order to fill that valence electron. So carbon will double bond with another carbon molecule. Wherever the double bond occurs, that region has like a kink, so it's slightly bent wherever that region is where carbon is double bonding to carbon, and thus why this part of the structure is slightly bent. So as a fatty acids, so fatty acid makes up the cell membrane. And remember we said that fats are non-polar, or lipids in general are non-polar, so they do not interact with polar molecules such as water. So they assemble as bilayers in water, and in which the tail part of it, so the tail is going to be hydrophobic. So the tail part, which are made up of fatty acids, do not like water, so they're going to be structured inwards. And the head region right here, which is the phosphate and the glycerol, this part right here is polar, so it interacts with water. So the head is the hydrophobic, so water loving, and the tails are the parts that are made up solely of fat, so they do not interact with water. All right, so steroids is our last type of lipids that we're going to be discussing, and this is the third one. So this is it. You can always identify steroids because they are the only types of lipids that have rings. Specifically, they have 4 fused rings. So this is what the chemical configuration of steroids look like. So if ever you're given this in a diagram, you should be able to quickly identify that these are steroids. Now, there are different types of steroids. You do not have to know every single type of steroid there is. But here's what you must know. So this is essential knowledge. There can be different types of steroids, or steroids can be placed into unique groups based on the functional groups or the unique chemical groups that are added to those steroids. So this is just general knowledge for you to know about steroids. There are different types, and what determines the different types are the functional groups or the unique groups that are attached to the rings, which give steroids different properties. You don't have to know any type of steroid or what functional groups are added to them or what properties that they will have. An example of steroid is testosterone. All right, so once you are seeing this slide, you know that we have come to the very end of this presentation, and also we're at the very end of Unit 1. All right, so let's quickly go over the essential knowledge that you should have for this unit, just basic stuff. There's a lot more detail than that we covered in the slides. So carbohydrate, what are the elements involved in carbohydrates? Carbon, hydrogen, oxygen. What are the monomers for carbohydrates? They're called monosaccharides, and the polymers are called polysaccharides. For proteins are made up of carbon, hydrogen, oxygen, nitrogen, sulfur. The monomer is amino acid, and the polymers are called polypeptides. Lipids are made up of elements such as carbon, hydrogen, oxygen, phosphorus, and we have like the phospholipids, if it's that type of lipid. And then the monomer is made up of glycerol and fatty acid, and there are no polymers. For nucleic acid is made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. And the monomer is nucleotides, and the polymer is DNA or RNA. All right, guys, we're at the very end of our video, and I hope that you guys are sufficiently prepared for your end of Unit 1 test, and I hope that you guys are sufficiently prepared for your AP Bio exam.